Right, welcome back. We'll just bounce through this super fast. You can stop and pause this as much as you want. I'm going to go through Dr. Clark's PowerPoint after this one, and we can hybridize the two, if that's alright with you. You'll get various bits from each one. Right, first up, muscles, yep, cells, force, great, 40% types. You need to know the three. Again, Clark Sheets are similar to this, and I'll run through his PowerPoints for you later. Fill those two lots of paperwork in. So cardiac, smooth, and skeletal. Cardiac obviously relates to the heart. Smooth is your intestines, iris, uterus, that kind of thing, glandular tissue, and then skeletal is obviously the voluntary stuff around your body. You can see that diagram there, that torso thing there. In terms of skeletal muscle, voluntary movement, blah, yep. Tendons attached to bone. Yeah, you can control it. Cardiac, yep. Rhythmically, lifespan, fatigue, myogenic, causes this impulse, yep, done, read all that. Small intestines, you say uterus, iris, things like that. Smooth, autonomic, so again you tend not to be able to control your small intestines or uterus, they literally have a mind of their own. Smooth muscle, often involuntary because not controlled consciously, yeah, done. I'm going to include that there. I have no idea why that suddenly appeared. So a little animation there. I don't want to worry too much about that, about the striated aspect to it. It's just visual. The ATP and contraction we'll come back to later on, but that's just again a quick recap. The bottom stuff here, this creatine, phosphate, we'll look at that, and again, or phosphocreatine, when we look at fast and slow twitch, so I will come back to that going forward. I don't want any of that for now. Yep, we'll come back to that more later. Movement, straightforward. Basic GCSE stuff, muscles pull, antagonistic pairs. Muscles can only contract and create a force. If you want to move a limb back, you need a muscle pulling in the opposite direction. If we click on this, I don't think it's going to work. Hey, there you go. Bicep contracts, you want to move your arm down. Tricep contracts, the other relaxes. Yeah, antagonistic pairs, all grand, all good. So again, we don't need too much of that. This next bit, if it's going to work, technology, don't just love it. Don't want that. We looked at this last time. Neuromuscular junction, this was the animation from previously, so I don't want to run through that. You can look back from the previous PowerPoint. We went through that. Sliding filament theory, now this is a bit I want to get onto. This is just a zoom, giving you an idea of the overview of the structure of this muscle. Again, it differs from Clark's, but I think it's useful because it's colourful and bright. So this is what I mean by a muscle, blah blah blah. So again, do make note of some of these here. I can send you pictures of these if you want to print some of those off, or do some screen caps. Within that muscle itself, we've got these bundles, groups of muscle fibers. So these are the muscle fibers which are surrounded by connective tissue. Yep, all nice and straightforward there. We're zooming in at a level now. So within this muscular bundle, this is a muscle fiber. So these are in effect your spaghettis. It says develop long multinucleated cells. So remember this muscle, you remember muscle cells? Many cells joined together. They share a common cytoplasm called a sarcoplasm, and these long fibers are found within. Moving down, we've got myofibrils. These are the individual ones which do the contracting. You'll notice that these banded patterns on them. You will learn what these are shortly in the next series of videos, talks. It's very namey. So again, nice and straightforward on that one. A sarcomere is basically a contractile unit. It's from this thin dark line to the next thin dark line. Sarcomere. It's this which will do the contracting. I'm going to get rid of you again because you're just doing my name. Going forward, if you go back to your colouring in diagrams that you've just done, you can actually mark a sarcomere on the pair of these things, and it will show you what the various bits are. So more of that later on. And then these protein filaments, this is the good bit with the animation that I like. Inside here we've got actin, myosin. These purple circles here, these are our, this is in effect what a sarcomere looks like in 3D, as it's contracts and relaxes in effect. Look at the distance between these two purple circles, these two Z discs or Z lines. Getting shorter, getting longer, getting shorter, getting longer. And this is where this idea of this sliding filament idea came from, in that it would appear that these fibres are sliding past one another. So more of that as we get to it. This little bit talks through the science behind it about these people you don't need to worry about, but it gives you these idea of lines and how it contracts. So again, do have a pause, do have a read, you get the idea. But very simply, these Z lines on here, you can see a Z line, a Z line. This collectively is a sarcomere, and inside you've got these two proteins. We've got the thick filament, which is myosin, and the thin one is actin. And the idea is that these two slide relative to one another and collectively cause the sarcomere to shorten and the muscle to contract. I'll run through this very briefly in terms of what you can see. We're going to come back to this in a bit more detail, but here's a Z line. Where's my pointer? I'm not sure why this isn't working up to this point. Here is a Z line, here is a Z line, and Z line to Z line, it tells us there's a sarcomere. Within that we've got these actins, we've got myosin. So what we're going to do, we're just going to click on a few of these things to get the notes from underneath to explain what it all is. So Z lines, define the ellipse of the sarcomere. Yeah, easy enough. Where's my pointer not going anyway? We'll work across actin, these are the thin filaments. Small protein subunits, basically two chain molecules, if you would. It's like a helix almost. Again, we're going to look in a bit more detail on that in a moment. Central one, we've got myosin. Myosin is the thick filament. Yeah, and that works with actin. This M line will not worry too much about for now. These other zones at the top, the I band, A band, and H zone, are important, and I'll explain why. The I band is a region that contains only actin. These next 2 or 3 slides are very useful. So that's our I band there, it's just actin. This region here, which is the A band, is a region which contains both actin and myosin. Yep, so actin and myosin would be our friend the A band, and this H zone is the region which is just myosin. So we have a region with just myosin, a region with just actin, and a region with actin and myosin. Okay. I'm not sure why this isn't working as it should, but there we go. Welcome to the jet age. Yeah, cross section. Oh, here's a sarcomere. What of 1 to 4 is that region? Is it line A, B, or C? Have a guess. Hopefully you've said it's got both molecules in it, the purple ones and the thing, so the myosin and the actin. So that hopefully should be B. Yeah, you got it right. And which region would contain the actin and the myosin? If you remember this region with just the actin, the thin filament, that was the I, so it isn't that. Z lines are these lines here, so it's not that. M line is the bit in the middle, it's not that. So we're basically going for A or H. I'm going to go for A because it is. There you go. More of this later on. At this point, what I want to do, I'm just going to bail out of this because it doesn't appear to be working quite as well as I would have hoped.